cruciferous vegetables had a protective effect against a variety of cancers. And they found then that broccoli was by far the most active. And as I looked, just sort of by chance, at the activity levels in some of these seedlings or sprouts, it sort of hit us in the head, because these broccoli sprouts had up to 100 times more sulforaphane or its precursor glucosinolates than market stage broccoli.
For the past 20 years, Dr. Fahey has explored the hypothesis of chemoprotection at Johns Hopkins University, with the idea that phytochemicals, plant compounds, can help guard against cellular damage that contributes to cancer and other chronic diseases. So his work, and he's known as the father of broccoli sprouts. And so I'm ready to just jump in and use every moment we have with Dr. Fahey. Do you mind if I call you Jed?
Doug, I don't. Please do.
Okay.
It's shorter and we're friends. I don't make my friends call me Dr. Fahey.
Okay. So let's just talk about broccoli sprouts. In terms of health benefits, why broccoli sprouts?
It's a great question, as are all your questions. I'll tell you that ahead of time. But why broccoli sprouts? Because they are loaded with a particular phytochemical called sulforaphane, or actually the precursor to sulforaphane. And this has been shown by us at Johns Hopkins and by many others around the world now to be one of the most potent, I was going to say action-packed, but one of the most potent phytochemicals in terms of its ability to enhance your health span or allow you to lead a healthy life longer and better until one day you just don't wake up. It attacks, or sulforaphane has been invoked or involved, shown to be involved in protecting against a large number of diseases due to a very unique mechanism that it has. But that's a long-winded answer to your question. We like the particular phytochemical. Broccoli sprouts taste good. If you like sprouts, you should like broccoli sprouts.
Terrific. And how did you arrive on the fact that broccoli sprouts were the king of these phytochemicals and the glucosinolates?
So in a sort of roundabout way, and I don't like, I hesitate to call anything, the king of phytochemicals. I don't like the word superfood, although that's used a lot, and I've used it too, in fact. We came about it because Paul Talalay and Yushin Zhang, his student at the time, doctoral student, in 1992 discovered sulforaphane in broccoli. And it was clear they had looked through a bunch of cruciferous vegetables with the idea that the epidemiologic evidence, from the 70s and 80s and early 90s showed that cruciferous vegetables had a protective effect against a variety of cancers. They were cancer doctors, they were cancer biologists. And at that time they said, okay, we've got this bit of evidence about cruciferous vegetables. Let's try to figure out why. Let's try to isolate the active component from some of these vegetables. And they found then that broccoli was by far the most active in some of their bioassays. This is, Petri plate work, as well as some mouse experiments. Knowing then that broccoli was the most active, this was in 1993, right after they published their discovery. It was in the New York Times. It was in the Proceedings of the National Academy of Sciences. Paul Talalay, my elder and mentor, was looking for someone that knew a bit about plants. I had come from the biotech industry. I'd actually done some work with broccoli, and I was looking for a change. So he lured me to Johns Hopkins, and I started looking all over for better broccoli. His challenge was, we found this compound in broccoli in tiny amounts, really tiny amounts. We need to find the broccoli that has most of it. We need to find better broccoli, if you will. To make a short story long, I then started growing it with cooperators all over the state of Maryland, where we were. But in the middle of the winter in Maryland, nobody grows broccoli. It's not like Wonder Valley in California, where you got good weather basically all year round. So I started looking in growth chambers in my lab, which was on the top floor of the hospital building, 13th floor of the hospital in the middle of East Baltimore. You don't do much rooftop gardening there, but you can grow stuff in incubators. And of course, I had to start small with seeds. And as I looked, just sort of by chance, at the activity levels in some of these seedlings or sprouts, it sort of hit us in the head. I said, whoa, what about broccoli sprouts? Because these broccoli sprouts had up to 100 times more sulforaphane or its precursor glucosinolates than market stage broccoli. So that was a sort of eureka moment. Then we started focusing on sprouts themselves with a vengeance. And of course, you don't need to go to the farm to do that.
Okay, that's really, outstanding that this mystery was revealed under those circumstances. And let's, for this lay audience, and myself, a lay person, can you help explain some of these key terms? What is sulforaphane?
Yep. So sulforaphane is just a small molecule called a phytochemical, meaning it's something that a plant produces, in this case, broccoli, and only a few other plants, not all cruciferous vegetables, as some people in the media have tried to represent. But it's a small molecule that is one of literally millions of small molecules plants make that help them in their environment, help them with reproduction, growth, and survival. Remember, plants don't have teeth and they can't run away from predators. So the way they fight back and try to keep from being eaten by bugs, mainly bugs and bacteria and fungi, is with phytochemicals. And these are compounds that deter attack by various predators or potential predators. Either they make the plant taste bitter, or they make it taste, any of a number of ways that a particular bug might not like, or they make it bactericidal, make it kill or disable bacteria that might want to colonize it. That's sort of why they're there. And I should also mention, they're the compounds that are involved in the color of plants, the greens and oranges and reds. And so those are all phytochemicals. We call them carotenoids or we call them anthocyanins, but those are all small molecules for reproduction, the scents, the smells that flowers exude, that may attract pollinators. All that stuff is done by phytochemicals. What they are not, I think, is even as important for the lay person to understand as what they are. What they are not is that they're not protein, fiber, carbohydrate, fat, DNA or RNA, or what major plant component am I missing? They're not the macronutrients that you think of usually as plants being rich in, and they're not minerals, which is the other, or vitamins. Those all are, there's a small group of each of those things, but there are 1 million or more phytochemicals. The estimates have been creeping up over the last 2 decades as detection and measurement methodology has gotten better and better.
What's the difference? What is a micronutrient and what is a macronutrient?
So a micronutrient is vitamins and minerals, basically. Okay, so there are a limited number of vitamins. We've all learned about them and somewhere in our high school classes, maybe. Vitamin A, vitamin B, all of the B vitamins, vitamin C, and so on. And then the minerals, iron, copper, magnesium, and so on. Again, there are a limited number that plants require, and there are a limited number that we require. If you look at the periodic table of elements, it's got, embarrassed, but I don't know how many, somewhere on the order of 100 elements. We don't need uranium, we don't need XY. There are a lot of elements we don't need, and neither do plants. They may take them up, they may be in there, and they may be toxic and contaminants to both people and plants. But so those are the microelements, vitamins and minerals. The macroelements are the big things that you find in plants. As I say, proteins, carbohydrates, and fats being the main ones. Carbohydrates are turned into fiber, so you can you can include fiber. I mean, those are really the macro elements. So if you look on the nutrition fact panel on the back of a food label, what you see listed typically is the macro elements: protein, fat, fiber, carbs. Calories, of course, are a measurement of energy. They're not an element, a macro element.
So we talked about sulforaphane. Can you talk about the glucosinolates as they pertain to making of sulforaphane?
Yeah. So I'm glad you brought up the word, because I can't be accused of using long chemical names exclusively. You get some of the blame. For a lay audience, there are very few terms I think that we really, really need to define. And I guess glucosinolates are one of them. So I talked about sulforaphane present in very, very low levels in broccoli. The way the plant makes sulforaphane, though, is it makes the precursor to sulforaphane, which is a slightly bigger molecule, also a phytochemical, though, and it's very benign. So the glucosinolates are a large family of phytochemicals. One of them is called glucoraphanin. Glucoraphanin makes sulforaphane. There are 120 or so glucosinolates, all present in cruciferous plants, the caper family, moringa, and the moringa family, and that's about it. There are a small number of plant families that have them. They are defense compounds because what happens, the glucosinolates are water soluble. They're present in vacuoles in the plant cell. So they're dissolved in the sap and the ooze and the protoplasm of the plant cell. There's an enzyme that's bound to membranes inside the same plant cells and in specialized cells. And that enzyme, called myrosinase, I introduced that term, is very active in turning glucosinolates into isothiocyanates. So those are the main families. Glucosinolates are exemplified in broccoli, especially by glucoraphanin, and myrosinase turns glucoraphanin into sulforaphane, which is an isothiocyanate. And sulforaphane is very active. It is where the rubber meets the road, what has the biological activity in both plants and humans. So why the hell would the plant go through all this trouble? Well, the reason it does is because the plant doesn't want this biologically active sulforaphane hanging around in its cells, because it will react with other organic material and sort of lose its oomph. But what it wants to do is, when a bug bites that plant, chews on it, then it wants the sulforaphane to be there. So what happens is when a bug, and this is the same thing happens with bacteria and fungi, when they break a plant cell in one of these plants, then the enzyme myrosinase and the water-soluble glucosinolate combine, because all of a sudden you got what was separate compartments, you got them all mushed together in a broken cell. And the sulforaphane is produced, and then in the case of some predators or some pathogens, they say, enough, I'm going away, and they stop feeding. It's a little bit weird because some glucosinolates are also feeding attractants to some insects, so they seek them out. So you have these pests that are specific to certain plants, like the cabbage looper or something like that, or tobacco hornworm, and they may actually be attracted to certain glucosinolates. That's probably not how evolution worked, but, well, I'm sorry, it is. So the insects evolved to like whatever, or to disregard whatever those phytochemicals were.
And when they say, so the plants, how is it that the plant's defense mechanism for its predators becomes beneficial for us as humans?
Well, it turns out that I guess repurpose is the best word, but when a compound like sulforaphane enters our body and is taken up, which it's rapidly taken up in the intestinal system, goes in the bloodstream, and it is taken up, it's absorbed, it enters cells fairly rapidly. When the cells see sulforaphane, they say, whoa, no good. this is a foreign compound. We're not used to seeing this. It doesn't do anything for us. Get it the hell out of here, literally. And so they have, in particular, human cells are loaded with, or many human cells are loaded with glutathione. That's it is the body's most predominant antioxidant. And so your cells that see sulforaphane gear up this protective response and crank out more antioxidants, more anti-inflammatory compounds. And so they mount this protective response. So, and then sulforaphane is ejected from the cell. It's literally given the boot on the back of some glutathione molecules. It's conjugated to glutathione, bound to glutathione, and thrown out in various ways. That happens very rapidly. And so sulforaphane, after you take, if you eat broccoli sprouts loaded with rich with sulforaphane, in very short order, when you measure sulforaphane excretion in your urine, which we've done more times than I care to remember, you see that sulforaphane is chucked out of the body very quickly. But if you then measure the protective response that is induced by that compound, it's long lasting, and it lasts for a matter of certainly many hours to many days, even on the order of a week or so in some cases. This is a complicated and confusing sort of discussion, or I'm making it confusing maybe. What happens is when the cells see sulforaphane and they crank up their protective response, they wind up inducing the production of a whole bunch of protective enzymes. Primarily, these are enzymes, hundreds. So they activate all these protective pathways, and those enzymes that are activated have different lifespans, if you will. We call it half-lives in science, but it means they stay around for hours to days, even weeks for some of them. And so that's why you can eat broccoli sprouts today and maybe not every day, maybe 2 or 3 days later. And as far as we understand, this is not absolute dogma, but as far as we understand, then you're going to achieve some degree of protection against a variety of conditions. By protection, I mean your antioxidant levels are cranked up or are enhanced. So you have more protection, for example, against air pollution injury. Your anti-inflammatory response is elevated. This may be effective against a number of neurological conditions, number of gastrointestinal conditions. The long and short of it is there are other mechanisms of protection. So we see protection against a variety of cancers in animal models, some evidence coming forth in human trials. We see protection against or some diminishment of the diabetic response. There are a whole host of conditions that we see responding to sulforaphane. Autism is another one I should mention. I mean, this is something I've been involved in a number of clinical studies on, and we see reduction of some of the symptoms. Let me just drone on for one more minute and say this sort of mechanism of action is quite different from the direct antioxidant response. We call sulforaphane an indirect antioxidant, but think about vitamin C, which a lot of people take as supplements, and of course we all know the stories about scurvy and how it was discovered and so on. When you take vitamin C, when you eat vitamin C, it's a strong antioxidant, but you pee it out almost immediately. I mean, it goes in, it does its protection, and it leaves the body. The story I spun for you about glucoraphane and sulforaphane is that they have a long-lived protective effect because of the mechanisms they upregulate, we call it.
How is sulforaphane used topically, like on skin? Like, how would that scenario work? Would you juice it? Would you make it into a puree by crushing it in a mortar and pestle?
Yeah. So we have done a number of experiments, published them, in which we have used applied sulforaphane to the skin. And we have seen effects in the sense that we saw an increase in some of these protective enzymes in the skin. biopsies, and we saw protection against sunburn, erythema. Erythema is the scientific term for it, but against reddening of the skin that's caused by sunlight or by UVB, specifically ultraviolet light, which is part of the sun. Of course, this is something you have to be a lot more concerned about in the desert in California than I do in mid-coast Maine. But topical application in the case of some of our experiments involved having sulforaphane in an organic solvent like acetone, not something you want to do as a matter of course. But this was a way of ensuring that it got in the skin, and we saw that it got in the skin. There are all sorts of other vehicles, of course, that can be used: oils, emollients, creams, etc. And we've done some of those experiments, and others have. So we know that sulforaphane can get into the skin. It's not duck soup. It's not really easy. And part of that, I think, is because the skin has this stratum corneum layer, this layer of dead cells on the top of it. We believe some of it binds and gets hung up there before it ever gets into the epithelial part of the skin, the living part of the skin that you really want it to be in. But anyway, so proof of principle, yes, it's protective when you apply it dermally, and we are working actively. I have a study ongoing. Well, we're just writing it up at Johns Hopkins on this, and there's a number of other studies that have been published. However, there's another way of getting it into the skin, and this is part of the study that we're writing up, and that's take it orally. So eating broccoli sprouts or taking a supplement with sulforaphane, we have found that the sulforaphane gets to the skin and is present there. So, it may not be that you have to apply it dermally. Of course, what we don't know a lot about is the levels you'd need to take orally to get, body-wide protection. Because when you're talking about protection against sunburn, you're talking about wanting to protect a large portion of your bod that's going to be exposed generally. But the other effect that we're hoping to be able to demonstrate some effect in is, I guess, most easily called skin aging or aging. The wrinkling and the movement away from a smooth baby soft skin to a Willie Nelson look. That thing called aging is something that you look around you see all sorts of people that age very, very differently. And so
you have beautiful skin.
Oh, thank you. So do you, man. I don't know if I can attribute it to sulforaphane, but I'm very conscious of this. I see people that are, I know are my age that have really smooth skin, and I see others, like I say, that look like Willie Nelson, who I love, but he's got some pretty rough, crinkly skin. So anyway, skin aging is certainly another application, and then sorry to interrupt, but then there are a number of skin conditions, diseases, so to speak, diseases or even genetic conditions that we and others have looked at. One of them is a very acute blistering. That's a genetic disease. Psoriasis, for example, and eczema. So these are all conditions that we're looking at, we or others are targeting. I should say others in that case. I don't have any ongoing studies in that area, but others are interested in it.
Have you heard of the solvent DMSO?
Sure. Oh, yeah. We use it all the time in the lab.
So how would you, could you make a tincture of broccoli sprout juice and DMSO and apply it topically?
Yes, you could. I wouldn't recommend it, though, because of the garlicky taste that you get in your mouth as soon as you put DMSO on your skin. Also, I'm not sure how wise it is to regularly use DMSO. I mean, in a one-off clinical study, it may be it's okay. I'm not aware that it's used regularly in cosmetics. I think because of the nature of it as a solvent. I mean, it's a pretty harsh organic solvent. It will dissolve a lot of stuff. And the other problem there is that the DMSO will go into your skin, no problem. It will go itself. It might still leave the sulforaphane behind. We looked, and we are looking at a number of oils. And as I say, some of the sort of commercial emollients and creams or serums that are designed or are have been characterized by others as allowing penetration of small molecules or drugs. We've found quite good efficacy with them. We've published a few trials in which we've used jojoba oil. Jojoba is a very commonly used oil in cosmetics, and it's interesting because different oils, jojoba, avocado, olive, MCT, all of the oils that you can think of that are typically used on skin, they're all different in the way they help vector sulforaphane in.
If you had to pick one, you would say jojoba.
I'd only say that for reasons that have to do with fatigue at talking about it for too long, we picked jojoba and it worked, 10 years ago. So, and we know that it's used in cosmetics. So, no, I'm not saying that's the best one, but it works.
What happens when you juice broccoli sprouts?
So when you juice them, you are that insect, that hornworm that I was talking about that was needed to be defended against. You're the caterpillar. So when you juice them, you break cells in the plant, and you break them massively. You break most of them, and then glucoraphanin is released into the juice. Myrosinase is available. The two things react, and you make sulforaphane very quickly. Happens within, seconds to minutes. And you probably, again, depending on the temperature and the conditions you use, probably almost all of the glucoraphanin is converted to sulforaphane within 10 or 15 minutes. If you do that, then my suggestion would be to drink that smoothie, drink that product, that juice, that homogenate soon, or freeze it immediately. And if you freeze it, you'll preserve most of the sulforaphane. If you drink it, you'll obviously get that benefit immediately. If you leave it around too long, or if you leave it in the refrigerator overnight, say, a lot of that sulforaphane is going to react with organic stuff that has been released from homogenizing the plant, from pureeing it. It may have some activity when you eat it, then when you drink it, but it'll certainly be reduced compared to what it would have been fresh.
It's a hard calculation to do, but if you were to take 8 ounces of broccoli sprouts, you would get about 5.5 ounces of broccoli sprout juice.
Yeah.
Pretty bitter. What kind of dose of sulforaphane would that be? And if you were sizing or specifying a dosage or recommendation of daily consumption of either the broccoli sprouts themselves or a juice, or perhaps, and I guess it's a similar reaction of what would happen in a blender if you were to take the 8 ounces of broccoli sprouts and blend them, but then you still have the insoluble fiber that the stomach has to extract the water molecules from, whereas the juicing is separating the water molecules from the cytoplasm, causing that reaction of the myrosinase and the glucoraphanin to form almost a liquid sulforaphane.
Correct. So that is more or less what will happen. I mean, I think, so you're going to get the fiber either way, whether you eat the sprouts, if I heard you correctly, whether you eat the sprouts fresh or whether you juice them and then drink them.
Well, I think if you juice them, you may get the water-soluble fiber. The insoluble fiber will be left in the pulp.
Sorry, if you leave them behind. And I'm confusing because I used to do juicing. I don't really do it anymore. But I was talking about making a smoothie, homogenizing. So yeah, if you get the juice, exactly. If you juice them, you're going to be making essentially an extract without the fiber, which is why I don't juice anymore. Because I prefer to have all the fiber, or the insoluble fiber as well as the soluble. But back to your original question, you said 8 ounces. Typically, I go for a 2 to 4 ounce broccoli sprout dose, and that gives a very, say 4 ounces, gives a very reasonable amount of sulforaphane. I mean, just for sake of calculations, which again, for a lay audience, they're not going to want to hear about moles and micromoles, but it's probably up about 500 micromoles from 4 ounces of glucoraphanin, which gets converted to sulforaphane. It's not all converted, maybe 400 micromoles of sulforaphane. But again, let's not get biochemistry gonzo here. A good dose would be come from 2 to 4 ounces of sprouts.
If you were to juice 4 ounces of broccoli sprouts, you would get a little bit less than 3 ounces, liquid ounces of broccoli sprout juice. With the water-soluble fiber, you would be removing almost all of the insoluble fiber. So I wouldn't recommend, doing that exclusively. But if you're eating sprouts and other plants, fruits and vegetables, the amount of fiber you're not getting in that is de minimis, but the concentrated, potentially bioavailable active sulforaphane makes, seems to me juicing the optimum way to get a dose of sulforaphane into the body in a very bioavailable, fresh way.
Yeah, I agree. What I don't know, and this, it's sort of an interesting question, which in a sense we've looked at in the lab, but when you juice and leave behind the fiber, there's probably going to be a substantial difference in how much sulforaphane is coming through in the juice, based on what I'm thinking is some of it may bind to the fiber that you leave behind. But I guess with a given juicer anyway, that timing is going to be pretty much the same no matter how you do it. So then it's going to be a question of how fast you drink it after you make the juice. So you should drink it fairly soon after you make the juice.
Well, I mean, I think in the hierarchy and the methods of creating this chemical reaction, you could have human chewing, and we know that humans do not chew effectively or completely, right? So you have chewing, then you have blending. Blending is pretty comprehensive, but then you're relying on the stomach to do the extraction, the separation of the water from the fiber, or juicing. So there's, many different ways of doing it. In the case of the juicing, I'm thinking cold press, slow juicing, maximum extraction, so that you're literally leaving the fiber so dry it would feel like wood chips.
Yeah. Let me back up and use my body as an example here. Just a minute. So when I think separating these into sort of 3 modalities is good. So when you chew broccoli sprouts, so right here, you're starting that enzymatic reaction to make sulforaphane. So that reaction's happening down to here. You get to the stomach, and the acid in the stomach is probably going to slow it down, but probably not stop it. And then it's going to go into the intestines and hang out there for a long time. If you blend it, you start the reaction, you're making a lot of sulforaphane, and that is going to go through and continue through into the intestines. If you juice it, so I guess the distinction I'm trying to make is the chewing method, you've got this enzymatic reaction happening all the way down probably into the intestines, whereas with the either blending or juicing, you put sulforaphane in basically, and it trucks right on through. It's not going to be deactivated by the acid in the stomach. But then don't forget that down here, you've got a huge amount of bacteria, and many of those bacteria families, in fact, all of us have bacteria that have myrosinase-like activity. And, that's important from the perspective that for example, if you were to I think it's gross, but if you were to cook broccoli sprouts, you kill the myrosinase in the broccoli sprouts. It's not available to you anymore until it gets to your intestines. Maybe the small intestines, there's controversy about that, but certainly in the large intestine, all those bacteria, you hear about good bacteria and bad bacteria. Well, you've got myrosinase containing bacteria also. And so they go to town on it, and they make And so they make sulforaphane in a different part of your body, and it's absorbed differently. It takes longer to get absorbed than when you start out by putting sulforaphane in here.
And how often do you recommend that people eat broccoli sprouts?
I suggest every few days, every 2 or 3 days, only because, you get tired of eating the same thing every day, right? I like to
I
mix
don't.
up.
I mean,
Well,
I don't, but
well, I guess I don't either. I eat my same oatmeal and chia seeds and teff and walnuts and blueberries every day. But later in the day, I like to vary it up. So
okay.
I think every 2 or 3 days is fine, but certainly you can eat it every day. You're not going to do damage to yourself.
What day or days do you recommend consuming the broccoli sprouts?
What do you mean?
Day 1, day 2, day 3, or day, if you're growing them fresh, day 3 and 7.
What day of growth? Yeah. We've in our research focused on day 3, on 3-day-old broccoli sprouts. But with a lot of seeds, I see when I home sprout, which I do all the time, usually the broccoli sprouts get better looking. There's a lot more mass after the 4th or 5th day. I think you have to treat it as a food and a medicine, and you have to not just focus entirely on how much sulforaphane you might be getting, but what's a satisfactory and fun food to eat. And so I think anywhere between 3 and 5 days. I generally don't grow my broccoli longer than 5, but
Jed, can you talk about how broccoli sprouts and sulforaphane may be protective against the common cold, virus, or flu?
Yeah. So there's a huge amount of anecdotal evidence from people who say that, they don't get flu, or they get less flu, or they don't get colds, or they get fewer colds when they eat a lot of broccoli sprouts. You have to discount a lot of that because it's uncontrolled experiments. However, there's a lot of stuff in the literature. There's a lot of work in the literature now in humans and in animal studies, mice studies, and in test tube studies showing that there is effect of sulforaphane against a number of quite a few families of viruses, including the common cold family. There's anti-replication, meaning keeping the virus from replicating. There's anti-infective activity, keeping the virus from infecting cells. And all of that is extremely supportive that, just like what you hear about vitamin C and the common cold and so on, that there is an effect of sulforaphane. I can't say that it's been borne out by large-scale clinical trials, in part because it's a phytochemical. It's not a drug. There's no big drug company funding these studies, but there definitely seems to be an effect.
And could you talk about, in the simplest form, how broccoli sprouts and sulforaphane affect the NRF2 pathway?
I was trying to keep away from too many technical terms, but you mentioned NRF2. NRF is a shorthand or an abbreviation for NRF2. And NRF2 is one of the master regulators in the body. It's a system that upregulates or induces or boosts a whole bunch of protective enzymes. And I was talking about it earlier, although I didn't use the term NRF2. But sulforaphane is the most potent naturally occurring activator of the NRF2 pathway that we know of. And this is based on now 25 plus years of data. And what it does is, so NRF2, it's called a transcription factor, and it basically tells the DNA to start making a certain enzyme, a certain product, which is usually an enzyme or a protein. And sulforaphane upregulates this pathway, and you get this coordinate upregulation of between 3 and 5% of the proteins that our body or that a cell makes. So it's a hugely important central biological regulator, and protective regulator by and large.
And when we talk about sprouts, do you believe that home sprouting is safe for human consumption?
I believe that home sprouting is safe for human consumption. Yes, I absolutely do. I've been doing it for years. I eat them. I give them to my granddaughters. depending upon their taste preferences of the day. And yes, I believe it's safe, and you have to pay attention to cleaning the seeds before you sprout. Other than that, you have a hygienic kitchen sink where you're chopping up chicken and fish and beef or something. But yes, if you pay attention to proper cleanliness, it is absolutely safe, I believe. And I believe it's safer than buying sprouts from the store, commercial sprouts, in large part because although commercial sprouters take proper efforts to keep their product clean, a lot of times sprouts in the grocery store stay on the shelf for forever until they start to get slimy and really lousy. And those are pretty gross. And, there's a gray area. When do they stop being really good and start to be potentially nasty?
Would homegrown sprouts, broccoli, alfalfa, mung bean, lentils, the like, would they be higher risk in your opinion or lower risk than buying regular market grade produce from a health food store or farmer's market?
Market grade produce, as in the big vegetables as opposed to the sprouts?
Yes.
I think that'd certainly be equivalent to that, or if not safer. I mean, remember that I think it's important for people who eat, in other words, everybody, to remember anytime you eat fresh fruits or vegetables, which is hopefully a lot, I'm leaving meats out of it because they're pretty bad microbially. But anytime you eat fresh fruits or vegetables, you're eating all sorts of bacteria that come along with them, even if you wash the surface or the skin. There's a huge amount of literature talking about endophytic bacteria and fungi. So whereas we used to think that plants were sterile on the inside, were totally devoid of bacteria, they're not. I mean, obviously there are some places in developing plant parts or organs that are free of bacteria, but there's a whole population of endophytic bacteria that actually go up the xylem and come down the phloem in plants. So you're eating a lot of bacteria when you eat fresh, washed, scrubbed fruit and vegetables, especially leafy vegetables. And sprouts are no different. There are some bacteria associated with them, but you do want to, when you make sprouts, you want to sanitize the seeds because you want to eliminate any potential human bacteria of human origin that may cause you harm. And I won't get into the details of that, but they're present on fruits and vegetables in the grocery store too.
Are you aware of any cases of hospitalizations, foodborne illness, or outbreaks, or even deaths from homegrown sprouting? I know you've studied the literature for 20 years.
25, 27, 28. Yeah. No, I'm not. Back in the 70s, 80s, 90s, there were a number of outbreaks in sprouts, but a lot of people then had really pretty nasty growing conditions and didn't pay attention to sanitation. But those were commercial sprouts. I'm not aware of homegrown sprouts causing problems. Could it happen occasionally? Sure, but I think not with people that are paying attention to proper sanitation and handling.
Terrific. Well, Jed, thank you so much for sharing this information. This was a deep dive into phytochemicals, glucoraphane, myrosinase, sulforaphane, with the father, of broccoli sprouts, Dr. Jed Fahey. Thank you very much.
Thank you so much for having me, Doug. If you wait too much longer, I'll be the grandfather of broccoli sprouts, but I'm glad you just called me the father of broccoli sprouts.
Okay.
Thank
Terrific.
you. Hey, thanks for watching. If you like this interview from the Commune podcast, then click subscribe and check out this video right here.